Okay, class. So these are the PowerPoint slides for the lecture two PowerPoints. I'm going to break this up into lessons, just like I did for the lecture one PowerPoints. So this set of PowerPoints goes over DNA and the nucleic acids. So just a little introduction. Scientists have been interested in the genetic material for a long time. Some of the evidence for this came from Darwin when he wrote On the Origin of Species, which was an attempt to explain how heritable traits can lead to variations in a population, like his finches with different beaks. Some had short beaks, and some had long beaks, and the long beaked ones were good at getting into cactus and stuff like that. And short beak ones had fatter beaks and they were good at, cracking nuts and stuff like that. So, Darwin was trying to explain how these traits can get passed on and that how these different species could evolve to have beaks that work best in their environment. And then we have Gregor Mendel, which we all know and love. Hopefully, we know him by now. And then 1869, we had the biochemist Michar, who discovered nucleic acids in bandages uh, inside white blood cells during the Crimean War, right? The Crimea, Russia thing. And then 1900s and 1926s, there were a lot of experiments that supported that chromosomes contain the genetic material, but uh, no one was really sure what a chromosome was. So this is a relatively recent thing we're going to talk about. And a lot of this, again, is just going to be review. All right. So problems with looking at chromosomes for our genetic material is that chromosomes are made up of both proteins and DNA. And so that has to do with packaging. And we're going to talk about this a little bit whenever we talk about how uh, meiosis and mitosis occurs later on. But the DNA is actually wound around these spools of histones. And we'll talk about what the histones are, but they're proteins. And it was really impossibly hard to know whether if it was when a chromosome was separated during mitosis or meiosis, and then that was the genetic material. It was really hard to know whether that was the genetic material was the protein, the histones, or the DNA. And in fact, we had no idea what DNA looked like or how it would function as genetic material. So, in fact, most scientists were team protein at this time, and the reason is that if you look at the alphabet of DNA, there's only four letters, and hopefully you guys know this. It's A and G and C and T. And in proteins, there is 20 letters, 20 different amino acids. I don't necessarily need you to know those for genetics class. You can look it up on a table. Um, so, what do you think would make a more complex language, right? Something that has the ability to make a human being, for example. Would it be something with four letters or something with 20 letters? And most people would agree that 20 letters is more can create more variation than four letters, and so that's why most people at the time believed that protein was the genetic material and not DNA. And in fact, not a lot of attention was paid to DNA at all until right after this Griffith experiment, which we're going to talk about now. So in 1928, Griffith performed four sets of experiments that helped determine, well, was meant to help determine what the genetic material was and how it worked. So this is one of the early experiments that led us down the path of figuring out what was the genetic material, either DNA or proteins. And so he took two different strains of Streptococcus pneumonia. So this is the bacteria that causes pneumonia. And generally, if you get it, then you would die. So in this case, there's actually two strains of bacteria. We have one. What are you doing? One that is. Hang on. My wrist is touching this pad and screwing it up. So I have one strain of bacteria. So here's the bacteria inside of here, and it's surrounded by a capsid. So the receptors are actually protected, and so the immune system has a hard time defending it. The other bacteria look like this, where their capsid is really rough, and so the receptors actually poke out, and so T cells or B cells or whatever can recognize this and then kill it. So this is protected from the immune system. And this is called S because it looked smooth under the microscope, and this is called the R strain because it looked rough under the microscope. So as you imagine, if your immune system cannot destroy this bacteria and it infects your lungs, then you die. So the smooth strain is the killer strain. The rough strain, if you get that, your immune system can detect it and destroy it. So with the rough strain, you live. Your immune system takes care of it. All right. So Griffith did some experiments, a series of experiments on this. And so the first thing he did was he injected what is all this junk here? I don't want this here. I can't see. All right. Well, anyway, so he injected living smooth strain bacteria into mice, and as you expect, they would die, right? Like we talked about previously. He also injected living rough cell strains of the pneumonia, and the mice lived, just like we would expect, because we talked about the immune system evasion previously. Now, so you guys know that if you heat stuff up for a long time, it generally kills it. Like if you're in the forest and you want to drink some dirty water, you could boil it for 10 minutes and that would pretty much kill everything that's in it. So that's what he did. He killed the S cells using a boiling technique, and then he injected it in the mice. So these are dead S cells, and the result was is that the mice, as you would expect, would live because it was injected with dead S cells. And then he did something very weird. So he took the dead S cell strain and mixed it with the living R rough strain and injected it in the mouse. And so you would kind of think that, okay, the living R strain didn't do anything, and the heat killed R S strain didn't do anything. So why would it matter if I mix them together? The mice should still live. But they didn't live, they died. And not only did they die, but when they took samples of blood, they found living S cells in the dead mice. Okay, so what's going on? And Griffiths was like, okay, well, there's really only two explanations. One is the bacteria, which were S strain, which were dead, were resurrected, right? They came back to life. And the other possibility is the genetic material that caused cells to be S was transferred to living, the dead S genetic material was transferred to living, so let's say dead S, and then living R, and that made them become living R. And so Griffith, being the scientist he is, postulated that it's probably the second thing that happened. And so he coined a few terms here. One of the reasons I'm telling you this is because he said that this was a transformation. So this is the word of the day. I want you guys, one of the words of the day. I want you to try to use it in a sentence. Transformation means that you've taken DNA out of one organism and put it into another, and that transforms that organism. So there's lots of examples of this. When I was in Singapore, they were looking at, let's see, let's open a new window. I'll be this person today. Come on. And we're going to grant to implement CRISPR-Cas9 into Bio 181. So that's what those emails are about. Anyway, all right, what were we doing? We're looking up stuff. So I was talking about Singapore. I was at the Genome Institute of Singapore doing breast cancer research. And while I was there, there were some grad students working on some zebrafish. So you probably know what zebrafish look like. And I'll just show you. So they look like zebras, right? They have white and black stripes on them. Here's a good example of zebrafish. So nothing surprising. And what they did was they took the DNA out of the zebrafish. Sorry, not out of the zebrafish. They took DNA out of a jellyfish, right? And they inserted that gene, which codes for a protein called green fluorescent protein. So if you excite this with a certain wavelength of light, it will emit a green color to your eye. So it glows green. So let's say GFP, because that's green fluorescent protein. And let's see if we get some images here. And so these are a lot of different fish, more than the green. But let's just say, let's look at these. So this is a green, this fish here, the zebrafish has green fluorescent protein, which was implanted during its embryonic germline, its single cell or very early on development phase, using gene editing techniques, and inserted that gene for green fluorescent protein. So when the fish grew up, all of its cells have green fluorescent protein and it glows green. This one has a gene from an amoeba, not an amoeba, anemone, right? I don't know why I wouldn't say that. Anemone, right? Which is red. And if you guys are in the life science department in the building, you can see an example of this coral in the saltwater tank that we have upstairs. So this term of taking DNA from one organism and putting it into another, Griffith coined as transformation. And we still use that today. So DNA from one organism and incorporating it into the DNA of another is transformation. That's the term. I'm sure I talk about that. So anyway, here's the where he talks about it, transforming. Griffith knew that the material was the genetic material, but he did not know what it was, right? Again, he's still stumped. We don't know if it's DNA or protein, but you're going to see this word transformation again. It occurs a lot in when we're doing genetically modified organisms. Just recently, I read that they were taking salmon DNA and put it in strawberries, to help make them more resistant to frost. So that would be a transformation. Okay. So that's Griffith. Make sure you guys know about Griffith's experiment. I'm going to ask you about it. I don't care about the year, but the reason I put the year is just you have an idea. So just so you know, the atomic bomb was detonated in 1945. So this was 8 years after the atomic bomb was detonated when Hershey and Chase wrote their paper and published it. And what they were doing is they were looking at viruses. So I just read a paper on this. I do a lot of reading, if you can't tell. And so what I was reading is that there are lots of strains of bacteria that people have gotten that are resistant to antibiotics. And one of these strains or group of bacteria is a super rare bacteria that's found in eye infections. And this woman had it in her ear. And she had her ear was bleeding, and I think she eventually lost her hearing in one ear, and it was spreading to another. And they gave her all kinds of antibiotics, and they couldn't get rid of this infection. And so what they did was they took these bacteriophages and they grew them up, because what's crazy about viruses, is that they're real specific for the host. So you wouldn't find like say a fungal virus in a human. It doesn't exist. You wouldn't find a cauliflower virus in a pig because they're too far apart. Like sometimes they can make leaps like from pigs or bats or whatever, but these are all mammals, right? So they're going from mammal to mammal. But generally, you're not going to get like kennel cough from a dog because your cells are quite different. And these phages, they're specific for bacteria. They only infect bacteria. So Hershey and Chase knew about these phages. They've been known about them for a long time. And they have two parts to them. So they have a coat that's made out of protein, and that's what's showing here, right? All of this part is protein, right? And inside here is DNA, and it's showing that it's being injected here. So that's kind of a spoiler alert. But anyway, what Hershey and Chase didn't know, and what was unique to viruses, is that viruses don't have chromosomes the way that we do. So virus chromosomes are the DNA is naked. My wife says I say that word weird. So DNA doesn't, no proteins are mixed in with it. It's just straight up DNA, and the DNA is found inside this phage. So Hershey and Chase obviously can't see these phages. This is an electron microscope of this. They really couldn't like visually look at this. The technology wasn't very good at the time. So they used a trick, which is to label with radiation. All right, so we have inside here, we have the DNA, and the outside part is protein. And so what they postulated is whatever was being injected in here, ignore the fact that this has already been told, whatever's being injected in here would be the genetic material. So here's the experiment Hershey and Chase did. They took a phage, right? And by the way, just so you know, we have there are even and odd phages. So there's T1, 3, 5, and so on, and then there's even, which are T2 and 4 and whatever. I'm not going to test you on that. I just want to let you to know. So this particular phage is a T2, but it doesn't matter. Just call it a bacteriophage. It's fine. So they took the phages and they grew them up in bacteria, right? So they were able to infect the bacteria, and they grew them up in two different radioactive substances. So in this case, batch one was incubated in radioactive S35. So this is the radioactive form of sulfur, and this is easily detectable, just like radiation or X-rays are detected by film. You could expose this to film and see if or you could use a Geiger counter or whatever. You would know if it's radioactive or not. So they knew that the phages, the protein part of the phages would be radioactive, and so if the protein was injected into the bacteria and they spun the bacteria down in a centrifuge, then all the bacteria would end up in the pellet, right? So the bacteria themselves would be in the pellet, and all the phages would be in the supernatant. And hopefully you remember the difference between the two. Anytime you run a centrifuge, you get two phases: the solid phase, which is called the pellet, and the liquid phase on top, which is called the supernatant. All right. So two phases here. We've spun this down, and the pellet contains only the bacteria, and the supernatant contains only the phages. And so in this case, the pellet was not radioactive. Now, you might ask yourself, well, what's the difference between the two? So there is no sulfur in DNA. It doesn't exist. This is formed by sulfide bonds that we're going to talk about in a minute, but that's why only sulfur is only found in protein. And then phosphorus is only found in DNA. So they use radioactive P32. It's only found in DNA. So they grew up these phages. This is drawn here in blue. And so again, they centrifuged it. They got a pellet. Ooh, I can move this. I want it to go away. How do I do that? Am I still recording? All right. Okay. Bye. All right. So anyway, that was annoying. Okay, we're talking about DNA, right? And so they centrifuged the viruses and bacteria, and then in this case the pellet, which remember contains the bacteria and the supernatant, and they found out the pellet is indeed radioactive. So this was the first evidence that DNA and not protein was the genetic material. And that's the Hershey and Chase experiment. It's super important for two reasons. One is, and I'm going to ask you this on a test, is phosphorus contained in protein? The answer is no. Is sulfur contained in DNA? The answer is no. Is carbon contained in DNA? Yes. Is carbon contained in protein? Yes. And I'm going to have you draw a couple nucleotides of DNA so that you can get the idea what the structure of DNA looks like on the problem sets. Okay, so that's their experiment. So that people were going crazy now because they're like, okay, tons of people were jumping off of the team protein and onto the team DNA after the Hershey and Chase experiment. And subsequent experiments showed that gametes only had half the amount of DNA as the organism that made them. The amount of DNA doubles during the cell cycle in mitosis. And in fact, prokaryotes and eukaryotes use DNA as their genetic material. The only exception is viruses, and viruses sometimes use single-stranded DNA or single-stranded RNA or double-stranded RNA, such as polio and HIV, right? And then we'll talk a little bit about prions. Prions are, or prions, depending on how you pronounce it, doesn't matter. Tomato, tomato, is protein, and that leads to mad cow disease. It actually, so prions actually interfere and change other proteins to resemble themselves, and in that case, they become non-functional. And this is generally caused by cannibalism. And you might ask yourself, well, how did cows eat other cows? I've never seen that before. And the answer is man did it. So man took these cow byproducts from the butcher, just like they do in dog food, and ground it up and fed it to the cows. So the cows unknowingly were eating other cows. And this is super common in like humans who do cannibalism generally in Polynesian islands. Anyway, so there's a little problem set on the new problem set for you to do a little experiment on something I made up, which is the honey badgers. I have been to Namibia, but I only saw one honey badger, and I didn't test its DNA to see what it was infected with. But you get a chance to do that on paper for the problem set that's coming up. Okay, so there's lots of evidence that DNA is the genetic material, right? And you know we know that DNA is a double-stranded helix. Watson and Crick did a lot of well, I'm not going to get on a high horse on that, but Watson and Crick basically stole the structure of DNA from Maurice Wilkins and Rosalind Franklin. Rosalind Franklin is the one that did most of the work, honestly, and she didn't get any credit for it. And it's kind of sad. But you could probably look this up on Google if you're interested in reading more about it. But it's totally a swindle. Like, if you took someone else's work and turned it in as your own, like in this class, what would happen? You'd get expelled, right? Because it's plagiarism. And they took someone else's work and published it, didn't give her any credit, and they got a Nobel Prize. Unbelievable. Okay, so anyway, nucleic acids involve DNA and RNA, right? This is deoxyribonucleic acid, and that's what DNA stands for, and ribonucleic acid, which is RNA. Genes are the heritable units, and they're composed of stretches of DNA, and we're going to talk about genes. DNA is a type of nucleic acid. It's deoxy. And so when we're talking about this annoying, where are you going? Over here. Oh, you can't even see it anymore. Perfect. So there are two main types of nucleic acids. They're based on their pentose sugar, right? So we have a 5 carbon sugar, 1, 2, 3, 4, 5, and then we number that clockwise from the oxygen. 1, 2, 3, 4, 5. So on the number 2 carbon, there's this hydroxyl group. On the number 3 carbon, there's also a hydroxyl group. And so this is ribose, right? It would be a nucleic acid if it had the base and the phosphate group, but this is just the sugar itself, and you can tell it ends in OSE, just like glucose or fructose or sucrose. And then here we have deoxyribose. So D, if I de-pantsed you, that would be take away your pants. So deoxyribose is taking away an oxygen. And you can see the only difference between this molecule and this molecule is on the number 2 carbon, there's a hydrogen. And on this number 2 carbon, there's a hydroxyl group. Other than that, they're exactly the same. But that doesn't mean there's not a big difference, right? Because on a molecular level, the shape of the molecule is super important. I mean, think about it this way. If you had a circle and you had a round peg and that went through a round hole and you glued something on it, like one atom, and it was exactly precise, it wouldn't fit anymore. So this, just because it's that small change, one atom, it's still a big deal. Next slide. Don't do that. Okay, fine. All right. So DNA, right? DNA is your set of instructions, right? Coded information that are found on genes, and that directs the cell's day-to-day activity. You can kind of think of it as a cookbook, right? A cookbook is your cookbook, like Joy of Cooking or whatever, is not going to have dinner ready for you when you get home, right? It's just a set of instructions. DNA is the same way. It's a set of instructions that's not involved in any way for doing anything. It's just an instruction book, right? The things that are responsible for reading the instruction book and cooking whatever the instructions are proteins, right? And the intermediary is RNA. In fact, one of the unique things that Watson and Crick came up with is DNA. Sorry, I meant Crick, not Crook, even though he's a crook. DNA to RNA to Freudian slip to protein. And this is called the central dogma. Central dogma of molecular biology is that DNA is the instruction set. The instruction set, you only have one set of DNA, one set of instructions. And so if anything happens to this instruction set, like let's say your grandma's secret recipe for, I don't know, banana muffins. So if someone went in there and erased it, then it would be gone forever. But if you made a copy of her grandma's recipe, then it wouldn't matter uh, what happened to that copy. What I like to tell my students is think of it as the Mona Lisa, right? So in the Mona Lisa, there's only one real actual copy of the Mona Lisa found in the Louvre in Paris. If you went and did something to that painting, they would like arrest you and throw you in jail for forever. You might even get killed. It's super protected, right? It's in argon gas and all kinds of junk. But you could go to the gift shop there and buy copies of it all day for like $10, even though the original is probably worth several $1,000,000. And that's kind of like DNA and RNA. So DNA is the original Mona Lisa, right? There's only one. There's no more. If it gets broken or whatever, then that's it. You wouldn't loan your friends that are going to go to a party your Mona Lisa, right? But you would be fine giving them a copy of it. Um, Same thing here. So RNA, DNA is in the nucleus. It never leaves the nucleus. RNA goes out to the cytoplasm C, where the ribosomes, and hopefully this is a review, ribosomes convert RNA into protein. And so converting DNA into RNA is called transcription, right? And the reason it's called transcription is because the letters in the language are similar. In DNA, there's an A and a G and a C and a T. And in RNA, there's A's and G's and C's. There's no T's. It's replaced by a close chemical cousin, which is U. And then in protein, there's 20 amino acids, right? Tryptophan, arginine, lysine, tyrosine. There's a bunch of them. So anyway, proteins are the things that are responsible for implementing the instructions of DNA. Proteins turn DNA into RNA. There's an enzyme, which is a protein, uh, called, it's DNA dependent, which means it depends on DNA, and it's an RNA polymerase. So it's an enzyme that makes polymers of RNA using DNA, right? DNA converted into RNA, and that's transcription. And then from transcription, the RNA gets processed, sent out to the cytoplasm C, uh, where the ribosomes are present. And again, ribosomes are proteins. And also, what's going on? What have I done? Ribosomes are proteins, and ribosomes are also contain ribosomal RNA, and we'll talk about that. It helps line up the RNA so that it's read properly by the ribosomes. And so those are what do what we call translation. And we're going to talk about that in the next lecture. DNA is interesting because it contains the actual directions for its own replication. DNA can't replicate it on its own. It has to make a protein that's responsible for copying it. DNA passes on exact copies of itself from generation to generation through mitosis, right? And then that's passed on to offspring through process of meiosis. And just remember, all cells in an organism contain the exact same set of instructions. So your brain cell and your heart cell and your lung cell, they're all the same DNA. It's just expressed differently. If you think about a television, right, a TV, there's only three colors. I mean, fancier ones might have a magenta, but generally it's red, green, blue, RGB. That doesn't mean that you can only watch red, green, and blue programs, right? Because each of the pixels are expressed differently. So the pattern of expression, genes being turned on and off in this case, creates the different picture or different cells in this case. So a heart cell would have different genes turned on and off than a brain cell. And that would have different genes turned on and off than, say, a lung cell, which is the exact same way that television is able to make all these different pictures by altering gene expression. And that's what makes cells different. Okay, RNA, we talked a little bit about this. When we talked about the DNA is turned into RNA through transcription, we're talking about messenger RNA. That's the message that comes from the DNA. So we sometimes put M in front of it, and that means it's messenger RNA. This is the main RNA that's going to be turned into protein. There's other kinds of RNA. They all come from DNA. There's tRNA, and that's inside of transfer RNA, which is responsible for bringing in the amino acids, right, during translation to the ribosome, and we'll talk about that. And there's an exercise in your problem set to do that. And then there's also ribosomal RNA. So tRNA, rRNA, and then mRNA. And then this ribosomal RNA is responsible for lighting up the guide of the messenger RNA into the ribosome. And there's some exercises for you to do in the problem set where you can get some practice on that. There's other functional RNAs. There's small nuclear RNAs or snRNAs. They are involved in post-transcriptional processing, which is part of a machinery called the spliceosome. And we're going to talk about this later on. I'm just going to mention it now. There's microRNAs, also called miRNAs. They regulate the amount of protein in eukaryotic cells. So there's a thing called RNA interference that was discovered in petunias on accident. And in fact, there's a Nova show on it. So if you guys look up Nova and RNA interference, you can watch kind of a 30-minute cool video on how this works and how they're using it to try to treat human diseases. But anyway, your own cell makes antisense RNA, which can interfere with protein synthesis in the same way that RNA interferences work as well. And then there's other things. These are also interfering RNAs, small interfering RNAs, and these protect against viruses and prevent the spread of transposons, which are transposable elements. And if we have time, we'll talk about these. I generally don't ever get to this because I get behind, but anyway, those are the different kinds of RNAs, right? So I might ask you about that. I'm probably not going to focus on the difference between micro and small interfering RNAs because basically their role is similar, very, very similar. But I certainly might ask you what ribosomal RNA or messenger RNA is, or siRNA, and what's their role in the cell. All right. So super important. These are the structures of the nucleotides, the bases, and there are two classes, and you're just going to have to learn these because this is how geneticists talk. So we talk about purines and pyrimides very different, especially when we're talking about mutations. So when we get to the chapter in mutation, it's going to be super important for you to know what this is. So you might as well learn it now. So a purine is something that has two rings, right? And if we look at DNA, I'm just going to quickly draw out DNA. So this is DNA. This is a hydrogen on number 2. Here's the base. So I'm not going to draw out every little structure on here, but so this would be the base, which we're talking about. This is the sugar. So this is the deoxyribose. I'm just going to call it D-ribose because I don't have enough room to write that. Here on number 3 carbon is the hydroxyl group. So this is number 2, number 3. Number 4 carbon, not much going on. And then the number 5 connects to the phosphate group. And then that connects to the next 3 prime over here. So we have the OH. This participates in the reaction. So this is number 3, number 2, hydrogen, and then this is the next nucleotide. And I'm going to have you guys draw out this structure just so you can sort of see what it is. So there's the base. This is the next nucleotide. I'll just call this, we'll call this a T. And we'll call this one G. So this would be T and G, right? And then we could have another one added here. And then we have the phosphate group here. I'm not going to draw the oxygens, but the phosphate group is here. And then the hydroxyl group is here on the 3 prime carbon, right? And the phosphate is on the 5 prime carbon. This is going to be important later on when we talk about the directionality of the DNA. But one thing that you can get used to doing to help you out is that remind yourself there's a phosphate on 5 prime, there's a hydroxyl on 3 prime. And I'll explain to you why that's important in a minute when we talk about replication. So purines have 2 rings that include adenine and guanine, right? They're very similar chemically to one another. They just have some different functional groups. Like here's a ketone. This is an amino group, another amino group. So they're very close chemical and size and shape wise. And then we have pyrimidines that consist of cytosine and uracil, which remember this is only found in RNA, and thymine, which is only found in DNA. Again, if we look at the difference between uracil and thymine, it's only this methyl group right here. So everything else is identical. That's why I say they're close chemical cousins. This one is also very similar. This is the amino group here that's different than these other groups. And so if you guys don't know the functional groups, like you didn't learn that or you forgot it, you probably should learn the functional groups because these are the ones that participate in the reactions. So the ones you should know are carboxyl, carbonyl or carbonyl, sulfur, or also called disulfide. You should know, and the carbonyls, you should know the difference between ketone and an aldehyde. And this is just basic review of functional groups. And then a hydroxyl group, and then a phosphate. Because we're going to talk about this later on when we talk about proteins. All right, so make sure you guys learn those functional groups. Anyway, so this is the different classes of purines and pyrimidines. You need to memorize this. I remember it as alphabetically U comes before Y, and alphabetically A comes before C. So I just remember that purines are A's and G's, and pyrimidines are all the other ones. It's relatively easy. This is what I want you to draw out, right? You're going to draw two strands of DNA. And so these are the terms, again, that you need to know. So this is a nucleotide. A nucleotide consists of three parts: the sugar, the base, and the phosphate group. This is negatively charged, and that's going to be important later on. And it's going to impart a negative charge to DNA. There's a lot of reasons to know that. A nucleoside is a term used for just the sugar in the base, not the phosphate group. So if you hear someone say nucleotide, you should think of all three of these. Nucleoside, just the sugar in the base. Right. The base is, so here's the number 1 carbon, number 2, number 3, number 4, number 5. Right. This is RNA because it has a hydroxyl group here. This is DNA because it only has a hydrogen. Now, something that you should have retained from intro biology and hopefully other bio classes is that when you want to make a macromolecule, and so the class of macromolecule, molecule, proteins, lipids, that includes all the fats, biological fats, nucleic acids, and carbohydrates. These are all considered biologically relevant macromolecules. And so if you want to make something, a macromolecule like DNA, you have to do what's called a dehydration reaction. And I'm not going to go into this, but you can go back and revisit like a basic intro biology textbook. What it means is that in order to build a molecule, stop doing that, build something, you have to remove water. Thus, dehydration. So you cannot build on this end of DNA, right? Because you have to make water, right? You have to take water out. So if you took this out, you only have an O, right? Where are you going to get the H, right? In order to build on DNA for an enzyme to build on it, it has to be able to have O's and H's because water is H2O. So this is the only end that nucleotides can get added. So DNA nucleotides can only be added on in the 5 prime, which is the phosphate, to 3 prime, which is the hydroxyl, right? Because we can add another one here. We just take out water. Direction, right? You cannot build in a 3 prime to 5 prime direction at all. And it's because of this dehydration reaction requirement, which is basic chemistry for macromolecules. All right. So central dogma, we talked about that. DNA is, and this is a eukaryote, right? It has a nucleus, eukaryote. DNA is made into RNA. RNA is made into a protein. That mRNA snakes out of the nuclear pore in the nucleus into the cytoplasm C. Super dangerous out there. Lots of nucleases that could chop up the RNA. So it's taking a super risk by leaving the nucleus, but it has to do it because ribosomes aren't found in the nucleus. And the ribosomes take the message on the mRNA, and they, using tRNA and rRNA, take and add amino acids to a growing chain, and we call that a polypeptide, which is also called a protein. Means the same thing, just like bucks and dollars or dinero, or cash and moolah, whatever. uh, bones. Okay, so central dogma I'll ask you about, and then here's macromolecules. So we're going to go over this. So macromolecules are polymers. Meros is part. So you have a monomer, that's a single part. A polymer is many parts. So if you want to make a polymer like sugars or lipids or long chains of DNA or RNA, then you have to take the individual pieces and link them together like beads on a string to make that bigger or macromolecule or polymer. So polymeros, many parts, monomeros, one part. All right. So condensation or dehydration reaction is the same exact word, right? I mean, not the same word, but it means the same thing. So that means you have to remove water to build a polymer, right? And that has to do with the hydroxyl group. And you guys are going to know these functional groups, right? The ones I wrote down. Uh, so the mechanism to break polymers is adding water, right? So we want to make to break down a polymer, like what goes on digesting your food in your stomach or your mouth. You add water, and that's hydrolysis, right? So using water to break apart. Lysis is break apart. All right. So yeah. So this is a little history about this, and we're going to talk about Chargaff's rule, and I have a little exercise for you in the problem set to do this. But in 1947, right before Hershey and Chase experiment, Chargaff already knew that DNA was made up of four nucleotides, A's and G's and C's and T's. And he was just kind of on his own studying this. Nobody was really interested in DNA at all. Um, and so one of the things he noticed is that all the organisms he looked at, they all contained DNA, right? So they're all had DNA, and not only that, but they all, um, except viruses, they all contained A, G's, C's, and T's in a specific order. The percent A always equaled the percent T, and the percent G always equaled the percent C. And this, my friends, is what helped Watson and Crick and Rosalind Franklin figure out the structure of DNA. So Rosalind Franklin was using X-ray crystallography. She was really good at it. I'm not going to test you on any of this. This is her picture. It's called photograph 51 because it's the 51st image she took. She later died. This is her. She later died from cancer, right? From all the radiation that this isn't like today where you go get dental x-rays or whatever. This is like so old school that they just kind of exposed radiation to crystallize DNA. By the way, Franklin was the only one that was able to crystallize DNA good enough to actually get a structure out of it. But they would crystallize the DNA and then they would expose it to x-rays. And they wouldn't just do it for a second like you do at the dentist. They push the button and it goes beep and you're done. This would be days and days of exposure to get this image. So she had this image, um, and she worked with this guy named Maurice Wilkins, who back in the day in the 50s, it was kind of like a good old boys club. They didn't believe women had a place in science. In fact, female faculty members couldn't eat in the same cafeteria as the male faculty members. So they were kind of excluded. It's kind of like the Masters Golf Club. And so anyway, Maurice Wilkins kind of thought he could do whatever he wanted with Rosalind Franklin. And when she went on vacation, he told Watson and Crick that they could come over and they would, they could have access to her drawer and look at her data. So they took the picture and they went back to their school and built models. You've probably seen those in your textbook or textbooks. And then they published a paper on the structure of DNA using Rosalind Franklin's crystallography data and the data from her notebook. Maurice, the reason we know this is because Maurice Wilkins wasn't mentioned in the paper either. And he was like, hey, I showed you guys, I let you into the building and showed you guys Rosalind Franklin's data, so I should get credit. And that started a big stink with the Nobel Committee, and then they figured out what was going on. So, anyway, this is just kind of my little soapbox, and I'm not going to carry on this too much, but I want you to see this. There's a commencement speech that James Watson gave. And he said there's a myth that Francis Crick, and I stole the structure from the people at King's. This is where Franklin and Wilkins worked. I was showing Rosalind Franklin's X-ray photograph, and woo, that was a helix. He didn't know that this picture was a helix. He was a biochemist. He would no way in hell would he know what this is. So that's I call BS on that one. And then a month later, we had the structure. What he doesn't tell you is that Rosalind Franklin, they called her because they couldn't get the structure right, and they asked her to come over and look at their structure, and she fixed it. She fixed their model using her data that they had already stolen that she didn't know about. So Wilkins should have never shown me the thing. I didn't go in the drawer and steal it. It was shown to me, and I was told the dimensions. What would you call that? It's called plagiarism, and they got a Nobel Prize for it. So anyway, the two crooks, Watson and Crick, sorry, came up with this structure of DNA, and we kind of went over it. So this is the number 1, 2, 3, 4, 5 carbon, which is this carbon here. 4 is in this ring structure. And then this is the base. The bases are bound here. And then the two strands are held together with hydrogen bonds. So there's 2 hydrogen bonds between A's and T's, 3 hydrogen bonds between G's and C's, as you can see here, 1, 2, 3. On the number 5 carbon is the phosphate group. It has a negative charge that's imparted on DNA. This is called the 5' end. This is called the 3' end, which is the only direction that this strand could copy. And it's this way, right? 5 to 3 on the 3. The other strand is flipped upside down. So here is the number. So let's do the numbering. So this is number 1, 2, 3, 4, and then this one right here is 5. And so this is the 5' end, and this is the 3' end. So that's what we call it anti-parallel. 5' to 3' on one side of the molecule, 3' to 5' on the other side. And it's because it's flipped upside down. This is what it looks like. This is the dimensions, 3.4 nanometers between turns, which are 10 nucleotides. Distance between nucleotides is 0.34 nanometers, which makes sense. And then the width of it is 2 nanometers. So 1 nanometer to the center. This is what we call the major groove because it's big. This is what we call the minor groove because it's small. Right? Major groove, minor groove. And you can see that here. Major groove and minor groove. All right. All right, different kinds of DNA. So we have a right-handed form of DNA, which is called D form, and that's bDNA, which is most common in all cells of your body. So aDNA is dehydrated form of bDNA. It's generally not found in cells unless they're dead because they're dehydrated. And then there's a left form, S form, which S stands for sinister because we all know that left-handed people are evil. I'm just kidding. Some of my kids are left-handed. Only the evil ones. Just kidding. Anyway, so we have the S or left-handed form of DNA, D dexterous, the right-handed form of DNA. zDNA is an interesting left-handed form of DNA. It creates a structure called, it's a zigzag. That's what Z stands for. So it's a zigzag structure. And this does occur occasionally in living cells, but we're not 100% sure what its function is, but it does seem to have a unique property. So that's why I mention it. I'm not going to test you on that. So you don't really need to know that. I'm just telling you there's different forms of DNA. Okay, let's look at the properties of DNA and RNA. So RNA is single-stranded, right? Whereas DNA is double-stranded. RNA contains uracil rather than thymine. It's super unstable, probably because think about a book, right? So if you left a book outside that was open, it would probably be a lot more damaged into the pages inside than if it was closed. So having the DNA molecule as a double helix, like a closed book, it's more protective. DNA, the two strands are complementary. That means that if A is on one strand, T is on the other. Has two hydrogen bonds. If C is on one strand, then G is on the other. This has three hydrogen bonds. It's anti-parallel. Um, so one end and this end, let's see what this end is. This is 5 prime and this is 3 prime. So this is 5, this is 3. If we have one that's 5 and one that's 3, then the other strand has to be the opposite because it's anti-parallel. And then this is 3 hydrogen bonds. So what I might ask you on the test, what is this, right? And so you know that A's and G's have 2 rings because it's making 3 hydrogen bonds. This has to be a G, and then therefore this has to be a C. Right? And you just know that by looking at the structure. Here's the phosphate group. Again, negative charges. A's and G's, C's and T's, because we're DNA, right? This would be U if it were RNA. All right. A and T, 2 hydrogen bonds, GC3, thymine, rather than uracil. It's very stable. Why? We talked about that. So it's moving right along. Prokaryotic DNA versus eukaryotic DNA. So prokaryotes have chromosomes, but they don't contain histones, so they're not mixed in with protein. Generally, they're circular and small. So it's like this, and they have a single origin of replication. Eukaryotic DNA is generally very long. So this is measured in, maybe 1 to 4 million nucleotides, whereas chromosomes are generally, I don't know, somewhere in the let's just say 20 million, 20 million range, 20 to 100 million nucleotides in a eukaryotic chromosome. I mean, this can vary. It really depends on what organism it is, but we're just speaking in generalities here. So prokaryotic DNA, small, circular. It's found in a region called the nucleoid, which, remember, prokaryotes is before nucleus, pro before karyote nucleus. They don't have a nucleus. U is true, karyote is nucleus. So eukaryotic DNA has a true nucleus. Usually these are linear, right? Although there's some exceptions. They're usually diploid, which means that, like if you're talking about ploidy, you have 2 chromosomes, one you got from mom and one you got from dad, and they just have alleles on them. And let's say that this isn't true, but let's just say that this is brown hair and this is blonde hair. So diploid means you have one copy of an allele from mom and one from dad. So di equals 2. And there's organisms that are octoploid, like strawberries, and that would, can you guess? Yes, 8 copies of their DNA. So they would have 8 chromosomes. These are called homologous chromosomes. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Too many, but anyway, you get the idea. Okay. Total combined for eukaryotes is usually in the billions of nucleotides. So 1 billion. That's adding all chromosomes together. All right. Eukaryotic chromosomes. So eukaryotic chromosomes are big, right? Like they're a little over 7 feet if you were to stretch it all out. And so there, that poses a big problem because you have to get 7 feet of DNA inside the nucleus of a cell that you can't even see with your naked eye. So that's a logistical problem that the cell solved by packaging the DNA on these chromosomes in a very specific way. This is accomplished by using proteins called histones that we talked about already, and the DNA is super tightly wound around these histones. So DNA plus histones is called chromatin, and chromatin makes chromosomes, right? And just a little interesting fun fact is that chroma is color and some is body. So chromosomes were actually named that because methylene blue stains chromosomes blue, right? So when they added methylene blue to it, they said, look at the colored bodies. And those ended up being called chromosomes. Chromosomes. All right. Any questions on that? So chromosomes are long stretches of chromatin. Chromatin simply means DNA wrapped around histones. This picture shows you the red are the histones and the blue is the DNA. All right, histones are abundant, small proteins. They have a positive charge. That makes sense, right? Because DNA is what's its charge? DNA is negative, right? So DNA is negative. Histones are positive. This is going to be important later on when we talk about epigenetics. But these are attracted to each other, so it's very easy to wind DNA around a histone, just like this. There's 4 parts to a histone. The round wheel, right? I like to think of it as like a kite string spool. There are 2 H2As, H2Bs, H3s, and H4s. So this is an octamer. Remember Meros's part, so it has 8 parts. And then there's another histone protein called H1 that can bind to the histones and wind them up. So this is representation is showing you that this is more tightly wound DNA than this. This is relaxed DNA, and this is wound DNA. So you got to kind of think of it as a scroll, right? If the scroll is open, then you're able to read it. And if the scroll is scrolled up, for the lack of a better word, then you can't read it. So your DNA is the same way. In order for your DNA to be turned into protein, it has to be able to be read. And so in eukaryotes with histones, you could control the gene expression by remodeling your chromatin. And we'll talk about that later on. But for right now, I want you to know about histones, the 5 parts of histones, what the charge on histones are, what their purpose is, and so on. I'm not going to ask you this specifically, but just this is so you know that each histone core, you need to know what the cores are made out of, right? So 2 of each of these. You don't need to know. It wraps around exactly 146 base pairs of DNA. This varies a little bit, and this is for humans, so it varies a little bit between organisms. There's a short stretch of DNA called the linker. You can see it here. This is the exposed part of DNA, not the coiled part. That linker in humans is about 38 to 65 base pairs, just to give you an idea of how big it is. And together this forms what we call the 30 nanometer form of chromatin, which is shown here. Now remember that this part of the function of histones is to package the chromosomes. So think about it this way. If you had a gym bag, and I don't know if you guys know this or not, but you have 23, so in humans we have 23, where's my pen? 23 pairs, right? Because we're diploid, so we have 2, 1 for each parent. So that's 46 total chromosomes. So imagine if you had 46 headphones in your gym bag and you just threw them in there. And then you wanted to pull out your favorite pair. I don't know. Or maybe that color of the week is, or day is red. So you're going to reach in there and try to pull this out. And what's going to happen? They're going to be all tangled up. They're going to be all twisted. And if you the harder you pull, chances are you might break it or whatever. So what's the best way to avoid this? Wind up your headphones before you throw them in your gym bag. So the cell is going to have to separate out not just 46 chromosomes, but during mitosis, that's going to double. So it's going to have 92 chromosomes, and it's going to have to send 46 into one cell and 46 in the other. Otherwise, it'll cause a mutation. So during meiosis and mitosis, the chromosomes are condensed about 10,000 times. So they're about 10,000 times shorter, about 400 times thicker. In the M phase, just to remind you, hopefully this is a review. So we do cell cycle phases, and I think we'll go over this again, but we have S phase and then G1, G0, M, which stands for mitosis and meiosis, and then G2 phase. So we're talking about during this phase, this is where condensation starts. Once you start condensing the chromosomes, you can't express the DNA. So it's kind of a big commitment of the cell because it's basically like you got to shut down everything. And then once you get through the M cycle, then you start reading the DNA and doing normal cell activities. So these chromosomes have to get separated during M phase into two daughter cells in mitosis of 46 chromosomes each. And if it didn't do this tight packaging, the chromosomes would certainly get tangled up and then they would get broken. And that leads to mutation. And we'll talk about whole chromosome mutations later on in the chapter where we're talking about chromosome abnormalities. All right, so this is, I don't care that you know this, right? But this is the typical form that you're talking about when we talk about 30 nanometer fibers of chromatin. These always appear as an X because you can only see chromosomes when they're condensed like that. If they're not in M phase, so they're not S, G2, G1, M, they're not tightly coiled or whatever. This is called interphase. This is M phase. So in interphase, you can't see the chromosomes. You can only see them after they've been replicated in S phase. That's why they always look like this X, because they're what we call sister, and hopefully this is a review, chromatids, right? This is a term. I'm going to go over briefly meiosis and mitosis, but you should be familiar with the term sister chromatid and homologous chromosomes already. All right, so packaging of prokaryotes is accomplished by a different method called supercoiling. And if you're in my biotech class, I have them run out pieces of DNA that are supercoiled and relaxed and cut. And anyway, so supercoiling just basically is taking like a rubber band and twisting it and twisting it. It's induced by an enzyme called DNA gyrase, which you probably should know. In other bacteria and in some eukaryotes, it's called topoisomerase. So gyrase and topoisomerase are the same thing. Well, here's a picture of relaxed DNA, and this is supercoiled DNA. So you can see it's kind of a prokaryote's way of packaging its DNA. Okay, so that brings us to the difference between remember that's true, and hetero is different. So euchromatin is chromatin that condenses and decondenses during the cell cycle, right? So we talked about the cells going through S phase and G2, and then it has to condense during M phase to separate the chromosomes. And then once it comes out, it decondenses. Because if it didn't, then the scrolls would always be shut and you wouldn't be able to make any gene products, so you would eventually die. Heterochromatin is chromatin that remains condensed in active DNA through the entire life of the cell. Um, and there's two kinds of heterochromatin. So there's facultative and constitutive. Facultative varies between cells. So in females, right? So this is something called dosage compensation. We have two chromosomes. Uh, Actually, I'm going to draw this a little different. We'll just draw it like this. We're going to call it the X chromosome. It doesn't look very X-y, but that's okay. So in males, there's only one X chromosome, and then we have a Y, which is very different, and it has SRY. We'll talk about this later. So in females, they get a double dose of the genes here, and that would be lethal because males here get one single dose from X, and this is normal. So to compensate for this, a female has to shut down either this chromosome by condensing it, just like we talked about with histones, or this one, right? And that process is a random process in humans. So either one of those could get condensed. And that's what we mean by it can vary between states of cells or even chromosomes. A bar body is the condensed X chromosome that you can see in females. So if you look under a microscope, remember I told you you can only see condensed chromosomes. So in the nucleus, you would see the condensed chromosome of a female. It's called a bar body, named after Dr. Body, who discovered it. Just kidding. I know you guys are digging my biology jokes. So, Bar Body, this dude named Bar saw this, and so he named it after himself because he was super modest. So, that's not found in males, right? It's only found in females because they have this dosage compensation where they have to condense one of their chromosomes. Constitutive is found in the same, in all cells at the same positions in homologous chromosomes. So telomeres and centromeres, and we'll talk about that later. But I mean, hopefully this is a review. But a centromere is the center point. Remember, centromeros. Meros means part. So centromere, mere, is the center part, and then telo means end, like telophase. And so telomeres are the ends of chromosomes. These are condensed across all of the same chromosomes. So chromosome number 1 has the same condensed DNA at the centromere, whether it came from mom or from dad. So this would be dad's number 1, mom's number 1. Centromeres are in the same place, and that's constitutive. So make sure you guys know the terms for all of this. All right, I just kind of I won't jump ahead, but centromeres are repetitive regions of DNA. This is where microtubules attach, in case you forgot. So here's the chromosomes. They're going to get separated. The microtubule organizing center builds microtubules that attach here, and then separates these into opposite sides of the new daughter cells. And this is during mitosis. Right, so that's what a centromere's purpose is. This occurs during M phase, and I'm not going to redraw the cell cycle, but you get it. Shortcut word for this is SYN, because scientists are lazy. They don't want to write all this stuff out. Telomeres are also repetitive DNA. They're at the end of the chromosomes. Conserved sequences, we'll talk about this and why it's important later on, but usually it's TTA GGG and that repeats over and over again. So and this has to do with uh, an issue during replication that we'll talk about. There's things called lines. These are long interspersed elements. Generally, this is repetitive DNA, but it's bigger. Usually, it's about 800 bases. It can be smaller, maybe 400. I mean, usually it's not 400, maybe 2,000 to 8,000. So these are big pieces of DNA that are repetitive over and over again. These are usually old viruses that contain like uh, genes that make gene products, like proteins. And so they can code for things like reverse transcriptase, which allows them to move around inside the cell. Um, all right. These are also known as transposons or transposable elements, but they're transposons because they're different transposons because they can transpose themselves. Whereas signs, they've lost the ability to do that, so they don't have the gene to make reverse transcriptase, and so they can be a lot shorter. Usually these are about 300 bases to 400 base pairs in length, and they're repetitive. In fact, 10% of all your DNA is signs from a virus called the ALU because this is a restriction enzyme that cuts it in a specific place. All right. So I'm going to stop here and then we'll pick this up DNA replication in the next lecture, which I plan on doing tomorrow. So hopefully that'll get posted then. If you have any questions, send me an email. Otherwise, you have a problem set due on in roughly 10 days. And then you have a quiz, but you don't need it. Hopefully you've already done the quiz. I think it was due yesterday for the first two lectures, and then there's another quiz that's due also on the 5th for replication, transcription, and translation. So any questions, email me. Otherwise, I will pick this up tomorrow.